So my name is Vivian Zhang. I'm the Business Development Associate at Omicsoft. And today we're glad to have Matt Newman, our Vice President of Business Development, to present our new function, GeneSet Analysis, which is integrated with our DiseaseLand and OncoLand. So for today's webinar, I will first turn to the presenter for a 30 to 35 minutes presentation, and we will hopefully to finish it in 40 to 45 minutes, following with the Q&A session. So please post any questions you have during the presentation. And after the presentation in the Q&A session, our presenter will pick up the questions and answer it for you. So if your question is not addressed or if you have any follow-up questions, we will certainly get back to you after the webinar. All right, so I think we will just get started soon with the presentation, and I will turn to the presenter. Welcome, Matt.
Okay, thanks, Vivian. As Vivian said, it should be about 25 to 30 minutes or 30 to 35 minutes, depending on how much I cover. I'm going to jump back and forth between the slides, and I will try to go slowly. I know, as most of you guys have seen these types of presentations before, you know I tend to go a little too fast, but it's going to be recorded, and hopefully that should help with everything. So we're going to talk about our new GeneSet Analysis software that's built into Array Suite, and just cover, just briefly what most of you guys know already, which is that Omicsoft has content, framework, and then a UI. In the framework, we have our land technology, we have the project management side of things, sample management side of things, the different analytics. In content, we of course have clinical data, genomic data, and we have our different lands. We have OncoLand, we have our DiseaseLand product, which includes ImmunoLand, CBM Land, as well as GeneticsLand. And of course our user interface, which has different parts to it. We have the land visualization, the genome browser, the project browser, the whole analytics side of things, and then 60+ different data views. So we've had a lot of requests to include a GeneSet analysis in our software, and the question is where to put it. So we've got the lands, and so our first thought was we should put it into the land side of things. And so in a previous version of our GeneSet analysis, everything sat inside the individual lands, and I'll show you that really quickly here. Here's my different lands. We have, I'm in the old ImmunoLand, and the way it worked before is that you had to add a GeneSet. So you would come in and you would add your particular GeneSet. You had to do it in this system. You had to come in and say add. You'd have your list of genes with your p-values and fold changes potentially. And then within each individual land, you had to go in and you had to use our gene analysis plot or table function. And so I would come in here and then I see all my different gene sets. I pick my gene set, and then it would give me the top hit. And in this case, it's showing IBD. Now, there were a number of problems with this. One is it was per land. So while I have this infliximab gene set in ImmunoLand, I might not have it in human disease land. So I come in here and I see my gene sets. It's actually empty. And so if I wanted to do the same thing in this land, I'd have to re-add it. On top of that, there was no concept of directionality. So we had p-values listed here, but there was no direction at all. I mean, it would tell you how many genes were up or down regulated, but that was about it. So we eliminated the land as the source point for excuse me. Take a drink. As the source point for the analysis. So the next question was whether to put it into the project side of things. And so initially we were thinking we already have the molecular signatures module. And so that's built into the microarray menu item here. And so I can still come in and go to microarray, go to annotation, and go to molecular signatures or David or gene ontology. And this works fine, but it's totally based on lists. So you have to have a preset number of lists. So I have a list of genes. I had to have an inference report that was based on microarray data, and I would come in and I would go to molecular signatures. I'd pick my organism and then run that, and it would give me back a set of top excuse me, a set of top pathways in the system. So that wasn't going to work. And so the end result was actually to put it into the project side of things, but also include both the land technology as well as those buckets from the molecular signatures database. So everything is run through the project side. There's a new integration menu. It runs on the server, so you have to have the server to make this work. It incorporates our molecular signatures database. It incorporates our land data. It has different atlas type data that I'll cover, and it includes all the different gene sets that you can enter. So I'm going to show that to you now. So I have a project open, GeneSet Analysis Demo 92816. Again, this is a server project, so you would create a server project beforehand. You would go into the integration menu, and you go to GeneSet Analysis. And so what it's going to ask me is to pick a universal set. Right now we're supporting human and mouse, but it's actually going to map them between the different species, and then a set of the different databases that you want to search. DiseaseLand covers our ImmunoLand and CVMLand databases. There's both a mouse and human version of that. Our molecular signature database for mouse and human. OncoLand, our reference database, and our new excuse me. And so after picking my databases, I can choose the type of data that I have. So I either have a list of identifiers, I have a list of directional identifiers, or I have a table that contains p-values, fold changes, and so forth. I can either have gene symbols or AFI IDs, ontrogene IDs, ensemble IDs, so forth. So we're going to do this, and we'll start with, I have a list of identifiers. We'll click Next. And I can either load this from a file, from a list that's in the solution, or I can cut and paste. So I'm just going to grab a list of genes here that I have that I want to analyze. We'll click Next. It's going to ask me for some cutoffs. If you wanted to return every single gene set, for instance, you could change this to 1, but by default it's 0.01. There's also a multiplicity adjustment. You can also test against the full target sets, and we'll go into that later, the details. I'll send that to everybody later. You can test against the full target set as an option. And then you can also publish these sets to a customer database. So I have an example customer database here, and we'll call this cancer-specific gene expression because I took some skin cancer genes. I'm going to give it a name and a project. And maybe a description. There's also a tagging concept that I won't get into right now, but you can tag them as well. And so we'll click Next again. It's got a list of 123 genes. We click Finish, and that's going to run on the server. And it should be pretty fast. Most analyses finish in around a minute to a minute and a half at the most. So while that's running, just cover the slides. Again, there's an easy-to-use wizard, and then we'll end up getting results. And I'll explain these results as this finishes. So that's done. If I have my email open and get an email, we go to the Analysis tab, click Update Project. That's going to finish. And so we get a new table here. And it's a pretty massive table in this case. It's got about 4,700 rows. I'll go into the table itself in a minute, but I always start with my overall top hits view here. And basically, this is showing me p-value on the x-axis, and on the y-axis it's showing me the top hits. And so you can see in this list that I'm hitting things like skin, as well as some other treatments that I'm not sure, I'm guessing are related to skin as well. The shaping is based on the type of list. So in this case, I didn't have any direction to my input list, but each one of these land-based lists has a direction. So for instance, this one are the up genes from the comparison of, this is a treated versus control, post-UV versus pre-UV skin, I guess it's a psoriasis project. So, you know, if you were looking for things that just correlated in the up direction, you could select those, for instance. Full means that it was the full list. So for each one of these gene sets, you're going to have a list of up genes, a list of down genes, and then a combination list. You can see that we've organized this into different categories. So, you know, overall top hits, there's a set of up and down that I'll cover later. Cancer-specific copy number, if you're looking for copy number changes, cancer-specific gene expression, and so this probably hits, should hit skin somewhere here. Yeah, list of 100 or so skin cancer genes. This is actually my list here, and so forth. Now, you know, you also get your list of treated versus control. So if there's particular treatments that you're trying to hit, you'll see that as well, and you'll see all the different lists here as well. Curated gene sets is going to come from molecular signature database. That's not going to have a direction, so you're going to see the purple circles here. What I tend to do with this analysis is go into my table report and just go full screen, sort by p-value, and then see what the top hits are. And you can see my top hit was actually from my own project. So I published this previously, and it's a list of 100 or so skin cancer genes. You can see that it will tell you the overlap, what the input gene set size was, what the target gene set size was, and then as you scroll over, it has all the different information, including a description and the name of the project and so forth, along with all the other metadata associated with that comparison. Now, obviously, there's going to be a whole lot of metadata associated with all the land sets, but if it's a customer-specific set, you know, like this one that I just generated, it's not going to have much unless I tagged it. The cool thing about this table is that it's clickable. So as you click on a row, you'll see that it automatically creates a Venn diagram view, and this is telling me that, you know, in my input gene set size of 123, 109 overlapped out of 123 with the target set here of a list of 100 or so skin cancer genes. Here's another one where we've got, you know, 123 is the input list, 38 overlapped, and then we have a p-value and so forth. If you want to see which genes are actually overlapped along with the full change of p-value, if it's available, you can do that. And you can also get a full list of the target genes. So if you wanted to see all the target genes, you could do that as well. All right, so that's the simple example. There's a question. Oh, and I think I just answered. They said for the Venn diagram and the details, what are the query and target genes? So the query genes are going to be your input list. So that's the list of that's this list of 123 genes that I put in here before, and the target genes are the individual gene sets. So, you know, for this particular gene set, we're looking at a comparison of treated versus control, post UV versus pre UV. All right. So that's the simple example. So let's get a little more complicated now. This time, I have a list of I have an inference report of full changes in p-values from my experiment, let's say. And so what I want to do is I want to take the top hits in this list and I want to put it into the GeneSet analysis. So we'll do the same thing. And again, we'll run this on everything except for the customer database. I'm going to choose the third option, which is that I have a table that contains p-values, fold changes, or other numeric columns. And it doesn't just have to be fold changes. You could easily have log2 ratios. You could have ranks. It doesn't really matter here for the type of test that we're doing. So we're going to go load from solution, choose my table, choose a fold change column, a value column, choose a rank column, and then choose some sort of cutoff. So let's say this is log2 fold change. Let's say greater than absolute value of 0.4, and then we'll say a p-value of less than 0.01. You can set how big this can be. So potentially this could be 5,000 genes. It doesn't tend to be as interesting when you do that. So I'm just going to set it to 500. It's going to do the mapping. Check your IDs. Again, we'll do the p-value, and then we're not going to publish it this time, but we could. Click next. You can see I hit 500 genes. Here's my list of genes, and then we'll click finish. And that's going to go ahead and run again on the server. And again, it should take, I would think, a minute or two at the most. You'll notice that it's running on the mouse lands as well. And so what it's doing is it's automatically mapping the genes from human over to mouse and then returning the mouse hits as well. And so while that's running, let's take a look at the lands and see what we're doing here. In DiseaseLand, for instance, you have, if I just look at the comparisons that are in DiseaseLand, you have a list of 2,300 comparisons, and each one of these comparisons is going to make up one of those gene sets. So, for instance, here's the early COPD versus normal. Here's treatment IL-23 versus none. Each one of these, we've stored the gene sets automatically inside the system. Additionally, for OncoLand, we've taken the top hits for cancer-specific expression from TCGA. We've also taken all of the normal tissue out of ImmunoLand and CBMLand and created a bucket of gene sets for that. And we've also done normal cell types from Blueprint as well as the single cell line land. So you're going to get all kinds of different gene sets that are already built in. All right, so that should be done. I would get an email, click update. And that downloads, and we get our new report, which is GeneSet Analysis Result 4. And so this time we're going to open up our overall top hits, and it's going to look slightly different. What you're going to see is that our direction is taken into account now. So imagine that you now have an input list and a target list. Each input list is going to have both a set of up genes and a set of down genes, and the target list is broken down into the up targets and the down targets and the full targets, which is the combination of both. So what you'll see is that my input list was related to infliximab, because you can see that the top hits here are showing both the full overlap, the up overlap, and the down overlap. For infliximab, week 30 response versus no response. So I'm clearly hitting this, and the reason I'm hitting it is I actually stole it from this particular experiment. And again, as you select something, so this is the downs, you're going to see the overlap. So the query had 286 genes in full. 150 of those were down and overlap with the target. Compare that to the ups. You've got, again, the input list had 214 in this case, and 143 of those are overlapped. And then the overall gene set, you're getting 179 out of 500 from the input list, and the target had only 193. So you're hitting a good portion of the target here. I see there was a couple questions. Where could I find these predefined gene sets from the disease experiments, these particular ones? They're only contained inside the GeneSet analysis, but I can provide you details of how we've done it. There's been some straight filtering that we've done, and there's a max number of genes that are included in each gene set. And then on top of that, we're going to do some calculations in order to figure out what the best size is for the gene set. And then there's a specific question about not seeing all the GeneSet analysis. I guess some people are so excited they're trying it while I'm talking. I'll shoot you guys an email afterwards, and we'll take a look. And then there's another question about what information about gene sets is stored other than the list of genes themselves. So for the land-based gene sets, you'll see that we're storing all of the metadata associated with that. So this is a really long description, but it's going to tell me that it's in IBD. The disease category was IBD. The state was colitis. The dosage, the platform. It was responder versus non-responder. It was expression analysis and so forth. This obviously will change depending on what you're picking. If we take a look at these results now, you'll see that I'm hitting infliximab as the response, but I'm also hitting some other treatments using infliximab, and then we can kind of work our way down the results to try to figure out what's going on here. One interesting thing is that you may be looking for things that are correlated, like the infliximab treatments. So you'll see, if we pick one of these here, this is the up. You can see it's going to tell me that it's up versus up and that it's correlated. But you might be looking for anti-correlation as well. So this one here, you've got the downs and the up. So this is actually down in my input list, but up in my target list. And you take a look at what this is, and it's actually a disease versus normal comparison for IBD. And infliximab is a treatment for IBD for some of the different diseases there. And so it's going to be anti-correlated. And we have a whole plot related to anti-correlated if you just wanted to get to your top hits. And as you select them, it'll bring up a table down here showing you everything. And again, all these different points on the plot are selectable. And as you select them, it's going to show you the Venn diagram of the overlap. Another question. Okay, is there an easy way to tell if the overall correlation with a gene set is a positive or negative correlation? It's a really good question, and it's not something that I have operational today. But what we're going to do in the next few weeks is essentially, in addition to the p-values that we're releasing, we're going to have a score, and the score is going to be a combination of the positive correlations and the anti-correlated. So this is just kind of an example that I set up yesterday for this. And again, this is not available yet, but should be in the next few weeks. Essentially, for each gene set that had direction, you can calculate a score. And for my input list here, what we see is that the top hits were actually for renal clear cell carcinoma from 3 different projects from OncoLand. And here's another one from TCGA. And these have really high positive scores as well as p-values. And then you also have the anti-correlation. So this other kidney type of cancer was actually in the opposite direction. So you're seeing an anti-correlation with that one. And I think that's going to answer that. But like I said, we should have that in the next week or two. Because I think it's an important question. Right now, what you have to do is kind of just filter for your in your table. So if you were looking for a particular comparison, or you found a particular comparison, like we can go to the top hits here, and we find, a particular description. What we could do is filter for that description here. So now we're just down to the 3, and then we can get an idea. In this case, you're not seeing any anti-correlation, and what that means is there's no overlap. So this one is completely correlated, both in the up direction and the down direction. The statistics, it's actually a Fisher exact test. I have the slide I'll go back to and show that. Maybe I can do that now. Yeah, so maybe I'll cover some of this since there's a bunch of questions about the criteria. Again, your input list can be anything, either a probe set, an oncogen ID, a gene symbol, and so forth. It can be a list of genes with p-value, with fold change, with copy number, expression difference, any way you want to rank it, essentially. And so what you end up with is an input list. The target lists from the lands, at least from our set, are based on a p-value of 0.05, absolute fold change of greater than 1.25, and then a maximum of 2,000 genes. And then it's ranked by p-value. We also have tissue-based expression and cell type expression. I haven't showed those plots, but those are the top 1,000 genes based on absolute excuse me. Those are based on the absolute difference of group mean from the global mean. And then, of course, you can have your customer-generated gene sets as well. The p-value, like I said, is a Fisher exact test. What's interesting, though, is how we determine the size of the target list. So when the input gene set is compared to a ranked target gene set, the maximal target gene set size is not always the best gene set size for calculating the overlapping Fisher exact test p-value. So we're going to use a method, and again, I'll send out these slides to everyone. We're going to use a method that's going to actually find the best cutting point. And so basically, we're going to end up with a smaller gene set size than the full set for each one of our different target lists. And that's why, if you guys remember, when I did the GeneSet analysis, just grab my list again here, you actually have the ability to change, to test against the full target set as opposed to the best cutting point. So this will go against everything if you choose to do so. Oh, somebody said they lost audio, so hopefully not for long. If your input list is 5,000 genes, how does it choose 500? There was a question about that. So if I had my list of genes again, and my input list was 5,000, let's say, you can choose the criteria. So fold change and p-value are going to be used to filter. So here I'm doing fold change. Here I'm doing p-value, and then I'm going to filter by it. So that's how I went from a couple thousand to 500. But it's totally up to you. You don't have to use this automatic filtering. You can just input the list. The one option I didn't choose yet is a list of directional identifiers. So if you've already figured out what the ups and the downs are, you actually get two boxes here, and so like you can have your obesity down list and obesity up list, and it'll do the same thing with direction. You just don't have the fold change in p-value. We'll let that run. while that's running, just talk about the roadmap a little bit. One missing point, I think, right now is integration with the lands. So what would be really nice is when you find, your list of comparisons here, this responder versus non-responder, up plus up, it's coming from the land. So, I could go in and I could manually search for this set of genes in GSD 23597. But it would be nice to have like a right click here where it would then take me to the into the lens and automatically filter it for either the input gene set or the target gene set, depending on what you were looking for, or the overlap. And so you can get an idea of, I can do that now by finding the interesting gene sets and then searching for them in the land. So for instance, this one here, if I go into one of the human lands, I can directly search for that comparison. And I can also search for the project itself if I wanted to look at it, the underlying expression data. But it'd be nice to have that automated and have it automatically take you into a heat map, for instance, or into the volcano plot in the particular comparison. So we have a number of ideas of what to do there. I think that's going to be over the next couple weeks as well. Right now, everything is based on genes, full genes. There's potential to add to our universal gene set list and include transcripts and potentially miRNAs as well. Additional visualizations, that's the volcano plot that I was showing with SCORE is one concept. And then right now we're just supporting mouse and human. The lands themselves, the land subscriptions themselves only have mouse and human data. But we've had some, I've shown this to a couple of our customers, and there's been some interest in the customers using their own gene sets, and they might have rat and monkey and some other species as well. So we'll support the automatic mapping for that as well. Let's go ahead and just take a look at the results of this one. And we're about maybe two to 3 minutes away from being done. So if you have additional questions, feel free to throw them into the question box. I think I've answered all the ones that have come in so far. But if we're happy to take questions for the next 10 minutes or so. So here's the result of that up and down list. Again, I have my overall top hits. This one's interesting, and this is something we're going to fix. It's just a visualization quirk. All of these have really low p-values. The p-value is essentially 0, 1 times 10 to the minus 325. But you can see it's hitting some areas that we haven't looked at yet, which is the tissue-specific gene expression and cell type-specific gene expression. So, for instance, you're seeing this one here is kidney-specific gene expression. It's hitting it from kidney. It makes sense because I took this list from a diabetes project where you were comparing you were comparing obese versus non-obese. So just keep in mind that you're not limited to just the disease versus normal and treatment comparisons. We also have tissue-specific and cell type-specific comparisons. And it's going to attempt to figure out which cell types there are. And it's doing this based on either all of the normal samples in DiseaseLand or the blueprint database. And so it's pulling from a number of different areas there. And that looks like we're done. Give it a minute to see if there's any questions. This is all available today. You probably need to talk to your. I would probably talk to your administrator or talk to us about updating your server because it's an area that we're developing on a regular basis. So you at least want to be up to date. As far as I would say at least from the last couple days. Could I show a T-cell specific gene set? I could. I would need a list of T-cell specific genes. I actually did this, and I didn't prepare for it for today, but I had a list of different cell types like T cells and B cells from a paper that I was looking at, and if you threw in the list, it does hit it hits T cells and B cells from the blueprint data set, as well as from some of the DiseaseLand data sets too. Another question, does the table report tab produce p-values of all tests? Yes, it does. When I ran the analysis, I set my cutoff as 0.01. You can set it to 1, and it'll return every single gene set in each direction, obviously, with the exception of gene sets that don't have any overlap. So if there's not a single gene overlap, even if you put a p-value of 1, it's not going to return that. All right, we'll give it another minute if there's any other questions. Like I said, I'll share the slides, or Vivian will share the slides later today probably, as well as the recording of this. And then feel free to shoot me an email, matt.newman@omicsoft.com, as well as support at omicsoft.com if you have any questions or you want to try to get this set up and working sooner than later. I didn't even show, actually I forgot, to show the management of the gene sets. So you can actually manage your own gene sets. So here's an example customer database. And you can see the different sets of genes that I've incorporated. Here's my skin cancer example genes, another one very similar to that. And these are based on the ones that I published earlier today. And it'll either, in some cases, it has the full changes in p-values, but only if you actually put it in there. And you can actually add in your own gene sets in this tab as well, either using the wizard, or if, let's say, you guys had your own set of 1,000 pre-calculated gene sets. There's a pretty simple format for it, and you can mass import these as well. So like here's essentially our format. It shows the gene set. You provide some tags and some metadata, and then you provide your list of genes along with the full changes and p-values that you're going to use, and it can be mass imported into the system. And the other nice thing is you can have more than one customer database. So if you wanted an oncology database and a clinical database, let's say, you could have those separate, and then you can choose to only run your analysis against those sets of data. All right, it doesn't look like we had any more questions. So I just want to say thanks to everybody, and if you have additional questions, like I said, shoot me an email.
All right, thanks, Matt, and thanks, everybody. I think we will post the handout and also the recording in a few days after the webinar. We'll send emails of the reminders to where you could find these resources, and certainly give the GeneSet analysis a try, or to contact us at any time if you have specific interests or recommendations for us. Well, thanks again for Matt for the excellent presentation and for all the questions from our audience. I think today we conclude today's webinar, and we will hopefully see you next time. All right, bye everyone.